Continuing the discussion on Western blotting, and we covered how to run an SDS page. And now that we've run the gel, what is the goal? The goal is to get the proteins out of a gel onto a surface where they're available for antibodies to bind to them. Currently, they're stuck inside of a gel, so antibodies will have difficulty reaching the proteins to bind to them. So we need to transfer these proteins to a membrane so that they're on this membrane and available for the antibodies to come in. So imagine that these little bumps are just protein, cellular protein that has been separated based on size, been transferred to this membrane, and now a specific antibody can come in and bind to our protein of interest, and only to our protein of interest, basically ignoring everything else. Now, what is the membrane that we are going to transfer to? The membrane that we're going to transfer to is called a PVDF membrane, which stands for polyvinyl iodine membrane. It is a very hydrophobic membrane. So the protein will simply be transferred over from the gel to this membrane, and since the membrane is very hydrophobic, the protein will just simply stick to the membrane. All the nonpolar amino acids will form van der Waals interactions with the membrane, and it will get that protein stuck to the membrane. It's going to be very hard to remove the proteins from the membrane at that point. So how do you transfer these proteins from the gel to the membrane? So we have to remember a couple of things. One was that our proteins were bathed and unfolded in SDS, and this gives them a negative charge. So SDS plus protein, we have a non-native unfolded protein that has a negative charge, because SDS has a negative charge, right? And we already separated the proteins based on size using SDS page. And to transfer the protein from our gel to the PVDF membrane, we're going to put the gel, here's our gel, we're just going to show a cross section. So the gel is in blue, our polyacrylamide gel. And we're going to stick the PVDF membrane right next to it. They're going to be touching each other. I'm not going to show it touching because it would kind of make this kind of impossible to draw, but they're going to be touching. There won't be a gap between the gel and the membrane. So this is our PVDF membrane. We're going to put this whole thing into a solution. Let me just draw it right here on the side. We know we're going to create this sort of sandwich of the polyacrylamide gel and PVDF, and we're going to stick it into a container. Here's my container. It's going to have a buffered solution in it. Here's my buffered solution, and here's my gel right next to the PVDF membrane. No gaps in between. The proteins are still in the polyacrylamide gel, and we want to transfer it over to the PVDF membrane. So what we do is that we create a positive charge on this side and a negative charge here, a voltage. And remembering that the proteins are associated with SDS, giving them a negative charge, the proteins will start to move in this direction from the gel toward the membrane. And once they reach the membrane, they will get stuck. Now, once they're stuck, you remove the membrane, the PVDF membrane, from the polyacrylamide gel, So here's my PVDF membrane. And all the protein that was on the polyacrylamide gel will be transferred over to the PVDF membrane. So now that smush of protein that I had that was separated based on size is on the PVDF membrane. Now somewhere in here is my protein of interest. What do I do to actually see my protein of interest and quantify the amount? And this is where antibodies come in. Our protein of interest, we're just going to call it PI. To get an antibody for our protein of interest, the protein of interest is going to be injected into some sort of animal to produce the antibody. So we take our protein of interest, inject into an animal, and we get our antibody versus our protein of interest. Okay? Now, typical animals that are used are mice, rabbits, and goats. So, when you're looking for an antibody versus your protein of interest, it'll have some sort of designation and etc. And so it'll be anti name of protein of interest, animal, antibody. What does this mean? Anti, for example, let's say my protein of interest, is MYC. MYC is a cellular protein. It's important in cancer biology. And the antibody against MYC was made in rabbits. So it'll be anti-MYC rabbit antibody. This information right here tells me the antibody is versus MYC. It will bind to MYC protein, and the antibody was made in rabbits, and it's a rabbit antibody. What you then do is you get a container, you put a buffered solution in here, and you put your PVDF membrane with the proteins into the buffered solution, and you add your antibody. Now, the first antibody you add is called the primary antibody. Or this antibody will bind to your protein of interest. So I have all this protein on the membrane right now. This is stuck to the membrane. I know I'm kind of giving it a little space between it. I'm just doing it for the drawing purposes. And my antibody versus my protein, let's say my protein of interest is right here. Antibody will go ahead and bind to this protein of interest. Okay? Multiple copies of it will bind. It depends. The more protein you have, the more antibody will bind. More protein will equal to more antibody binding. After you introduce the primary antibody into your solution, the next thing you want to do is you want to remove any free-floating antibody out of here before the next step. Because you're going to have some antibody that hasn't been bound to your protein of interest because there's no more binding sites available. And sometimes, depending on the concentration that you're using of antibody, you might get some non-specific binding happening to other proteins, but it's rather a weak binding. So you want to dissociate all these antibodies that are not bound to your protein of interest. Okay? So you do wash steps 3 times. And all this is that you take the container, you change the solution, and you put some fresh solution in there that doesn't have the antibody. But you have your PVDF membrane in here with all the protein still stuck to it. And your protein of interest was right here. and you have your primary antibody bound to the protein of interest. But you removed all the other extra primary antibody that was sort of floating around or maybe non-specifically binding to other proteins. Those have been removed. After this, you have to put on the secondary antibody. Now, what is the secondary antibody? So we have to remember what the shape of an antibody was. We saw this previously, but basically we had this Y-shaped protein, right? And you had the variable regions right on the tip. These are the sites right here that bind to your protein of interest, what the antibody actually reacts to, binds to. And then you had the Fc region. The Fc region is going to be unique depending on the organism the antibody was made in. So a mouse antibody will have a different Fc region in all of its antibodies versus, for example, a rabbit antibody. Here's my primary antibody. And we said that it was, well, we actually didn't say what it was, but let's say our primary antibody is a rabbit antibody. The primary antibody was made in rabbits. So at this point, what will happen is to make a secondary antibody, this Fc region right here, just the Fc region, is introduced into another animal. So we take the Fc region from rabbit, we put it into, for example, mice or a mouse, and we're going to produce antibodies versus rabbit Fc. Now that's a little bit to take in, but it's nothing different than what we talked about before. If I'm putting a foreign protein, which is the Fc region of a rabbit, into a mouse, I'm going to get antibodies versus that Fc region. So what I now have is, and let's just change the color. This antibody will say it's green. Okay, so here is my original antibody, which was against my protein of interest, my primary antibody, which was rabbit. What will happen if I introduce now the antibody versus the rabbit Fc, or anti-rabbit? So this would be anti-rabbit antibody. Mouse antibody, because it was risen in mouse, right? So if I put this antibody in, well, this antibody, its variable region, will bind to the Fc region of our primary antibody. Now you might be saying, okay, well, that's neat, but what's the point? Well, the thing about secondary antibodies is once you have isolated the secondary antibody, for example, anti-rabbit mouse antibody, what then happens is that you do a little bit of genetic engineering. And the genetic engineering that's done is that an enzymatic domain is added to the Fc region of the secondary antibody. So we're going to put an enzymatic region. Now the typical enzymatic protein that is used, domain that is used, is HRP, which is horseradish peroxidase. The most important thing is that this is a peroxidase. And the reaction that it will catalyze is one where ROR prime plus 2 electrons from a donor molecule plus 2H plus gives me, and this is peroxidase being the enzyme, gives me ROH plus R prime OH. Now, again, that is nice, but now that I have a peroxidase enzyme attached to my secondary antibody, I can put in a substrate. Once the peroxidase catalyzes the reaction on the substrate, one of the outcomes of the reaction will be light. Here you see an example of HRP, horseradish peroxidase, working on a specific substrate, and once the substrate, the reaction occurs, one of the products, or one of the outcomes, it's not really a product, is light. That's the energy released by the reaction in the form of light. So going back to where we were at previously, before we started talking about all this, is that we had our PVDF membrane in a solution, bunch of proteins, right? And we had our protein of interest somewhere here, and we had our primary antibody, which we had colored in red, already bound. Of course, you'll have more primary antibody bound the more of the protein of interest you have. Now I'm going to add my secondary antibody, plus when I add my secondary antibody, I will have although the proteins are already still attached to the PVDF membrane, my protein of interest, my primary antibody, and now a little bit of a problem. Let's just raise the level of this solution a little bit. Now I'll have my secondary antibody attached to the primary antibody, which has an HRP domain, right? Horseradish peroxidase. So this is going to be secondary antibody has HRP domain. Of course, you're going to have some free-floating antibody here too, and you might get some non-specific binding, and you do the same thing as we did before, which is we do a 3 times wash step. That is simply you're going to remove the solution that is bathing the PVDF membrane, and then put in fresh solution that doesn't have antibody, and you're going to do that 3 times to remove any non-specifically bound antibody, or any antibody that is in solution is not bound anywhere at all. And you end up with your PVDF membrane. Now, of course, this is in a solution, but we're not going to draw the solution. We have our protein of interest. We're not really interested in any of the proteins. Let's just look at our protein of interest. We know that the other proteins still exist. We have not removed them. But let's just concentrate on our protein of interest. And we have our primary antibody bound. We have our secondary antibody bound to the primary antibody with an HRP domain. Now what you do at this point is you add the substrate for HRP. The reaction will lead to light, right? The energy from the reaction, because it's an exothermic reaction, the energy that's released by the reaction will be in the form of light. Now, you add the substrate, and then you put an undeveloped film on top of the PVDF membrane. Then you develop the film. Because once light hits that undeveloped film, it will create a band on the film once the film is developed. And you get something that looks like this. This is an example from a research paper of a Western blot. Now, each individual box here is a different Western blot. So this is one Western blot, this is one Western blot, this is another one, this is another one, this is another one, etc., etc., etc. Okay? Now, for those of you that are working on your flaviviruses, on your papers, and know something about flaviviruses or viruses in the Flaviviridi family, these are all viral proteins. These cells have been infected with hepatitis C virus. And when it says core, for example, here, that means the primary antibody was versus the core protein of the virus. E2, the antibody was versus E2, and S2, the antibody was versus non-structural protein 2, etc., etc., etc. Now, these are just on top, these are just different treatment groups. We're not going to really get into this, what that means and what these treatment groups are. To do that, you would have to read the paper and look at it more carefully. But the main thing is that between the treatment groups, for example, you can see that the core protein is not being shown in the bound fraction. So here you can see right here, there is no core protein. Whereas in the total, there is core protein. And you can see between the plus and the minus, in the plus, there's less core protein than in the minus, right? Because the band is thicker, bigger in the minus. That's why it's quantitative. And why is that? Well, if I have more protein, more primary antibody can bind, right? So more protein of interest, more primary antibody can bind. If more primary antibody can bind, then more secondary antibody can bind to the primary antibody. And that means more HRP activity. So when I add my substrate, I will get more light being formed. And if I have more light, I would have thicker bands. Okay, so let's see, is there another good example that really showcases this? I think, for example, you can look at NS2 right here. Let me get rid of this. And you can see that, for example, NS2, that there's even a slight difference in size of NS2 from the total plus and the total minus. Why that is the case, I'm completely unsure of right now, but I would have to look at the paper a little bit more closely. And you can see that NS2 in the bound plus, it's a pretty big band. There's a lot of protein there. Another example could be, we can look at, for example, the core protein here. You can see in the total plus and minus about the same amount of protein, but none in the bound fractions. And these are all separate Western blots. So Western blotting is a tool that is still used widely, where you can quantify, because you can actually quantify the size of these bands to tell you about the percentage change between your maybe treated or untreated and your controls as far as the amount of protein that is being shown on your Western blot. So we had two assays that we talked about. We had reverse transcriptase, real-time PCR, and this is a two-step process where you get your messenger RNA, you do reverse transcription to get your cDNA library, so this is to get your cDNA library, and then you quantify the amount of your original messenger RNA by doing real-time PCR. So here, we're quantifying the amount of a messenger RNA, a specific messenger RNA. And we have Western blotting. Which will quantify the amount of a specific protein. So a mRNA, a protein, again, specific. So in the whole molecular biology of gene expression, where you go from DNA to RNA to protein, we can find out the amount of RNA or messenger RNA and the amount of protein using these two different methods. One is reverse transcriptase real-time PCR, or RT-qPCR is sometimes how it's abbreviated, RT-qPCR for quantitative PCR, and Western blotting for the amount of protein. The question that might come up is, well, if I know the amount of messenger RNA, don't I know the amount of protein? And that is not true, because the amount of messenger RNA tells you the amount of messenger RNA. Sometimes, in some cases, for some proteins, the amount of messenger RNA is indicative of the amount of protein that you will have from that gene. And in other cases, the amount of messenger RNA is not indicative of the amount of protein. Okay, so if you're trying to quantify the amount of a specific gene's product, if the gene's product is a protein, you would have to actually do Western blotting to have that information. Okay, we'll stop here and I'll continue later. Okay, thank you guys.